Welcome to Mr. Chalk's Revision Tips. In this video we will look at the control of the cell cycle, the stages in the cell cycle, and what the actual purpose of the cell cycle is. When you look through your notes, might also notice some points where it says check questions. You need to go and have a go at these questions. So the cell cycle is a four-stage process consisting of gap stages, synthesis stages, and mitosis. So like I've just said, it's split up into stages. There is some sort of biochemical control that is to do with certain proteins called cyclic dependent kinases being activated or not activated at certain points. And the purpose of it is to go and produce cells when they need it and ensure that cells are produced so that there is no mistakes in the DNA so they do not become cancerous, for instance. So the cell cycle is split up into stages that the cell passes through as it's getting ready to replicate. So there are three main stages. So you've got an interphase, and the other key thing is the two main proteins called cyclins that control the cell cycle. So you've got cyclic dependent kinases. So these are responsible for the progression or the progress of the cell through the various checkpoints. And so cyclins regulate the cell cycle and control when certain aspects take place. So the first part of it is called the G1. So this is also called the gap phase or gap phase 1. So the cell grows physically larger, copies organelles, and makes the molecular building blocks that it needs for later steps. In the S phase, the cell synthesizes a complete copy of its DNA in the nucleus. It also duplicates a microtubule organizing structure called a centrosome. So the centrosomes help the DNA separate during M phase. During the second gap phase, or G2, the cell grows some more, makes proteins and organelles, and begins to reorganize its contents in preparation for mitosis. During the mitotic, or M phase, the cell divides its copies of DNA and the cytoplasm to make two new cells. So M phase involves two distinct division-related processes. And in cytokinesis, the cytoplasm of the cell is split into two, making two new cells. Cytokinesis usually begins just as mitosis is ending, with very, very little overlap. The G0 phase is a period in the cell cycle in which the cell exists in a quiescent state. So that's in a state where it's not dividing. The G0 phase is viewed either as an extended G1 phase, where the cell is neither dividing or preparing to divide, or just as a distinct quiescent stage that occurs outside of the cell cycle. At the heart of the cell cycle control system is a family of protein kinases, known as cyclic dependent kinases. The activity of these kinases raises and falls as the cell progresses through the cell cycle. The most important of these proteins are known as cyclins. So the G1 checkpoint is where you carry out typically arrest cell division if environmental conditions make cell division impossible, or if the cell passes into G0 for an extended period of time. At this point, the following checks are carried out. So it checks for cell size, nutrients in the cell, growth factors, and most importantly, if there are any damage to the DNA. The G2 checkpoint is important in the cell cycle. So the checkpoint ensures that the cells don't initiate mitosis before they have a chance to repair any damaged DNA. The spindle fibre or the spindle assembly checkpoint is a checkpoint ensuring that chromosome segregation is correct. The spindle checkpoint or mitotic checkpoint is important to happen before mitosis. So during mitosis or meiosis, the spindle checkpoint prevents anaphase, which is the onset until the chromosomes are properly attached to the spindle. And in future lessons, we will look at the process of mitosis. So a CDK is an enzyme that acts negatively or acts on negatively charged phosphate groups to molecules in a process called phosphorylation. Through phosphorylation, CDKs signal the cell that is ready to pass into its next phase. So cycling is named such because they undergo a constant cycle of synthesis and degradation during the cell division. When cyclin is synthesized, they act as an activating protein and bind to CDKs, forming a cyclin CDK complex. This complex acts as a signal to the cell to pass into the next phase of the cell cycle. Eventually, the cyclin degrades, deactivating the CDK, so signaling the exit from that particular phase. G1 cyclins bind CDK proteins during G1. Once bound and activated, the CDK signals the cell's exit from G1 and entry into the S phase. When the cell reaches an appropriate size and the cellular environment is correct for the DNA replication to take place, the cyclins begin to degrade, so to break down. So G1 cyclin degradation deactivates the CDK and leads to the entry into the S phase. Mitotic cyclins accumulate gradually during G2. Once they reach a high enough concentration, they can bind to the CDKs. When mitotic cyclins bind to CDKs in G2, the resulting complex is known as mitosis promoting factor. This complex acts as signal for the G2 cell to enter mitosis. Once the mitotic cyclin degrades, the MPF is inactivated and the cell exits mitosis by dividing and re-entering the G1 phase. Thanks for watching.